Okay, so I'm going to talk to you guys about the early detection of non-native species through the use of environmental DNA. So this work kind of encompasses two of the work packages, work package 4, which was done before I joined. And the aims of this were to test, design and test methods for early detection of non-native species using PCR and meta-barcoding in marine coast. And the outputs for this were a qPCR assay for Irish strains of Didymnum vexillum. And onto work package 8, we have two and outputs, the first of which is to produce a DNA barcoding database for non-native and invasive species, Irish Sea, testing multiple sets of barcoding primers, and to test each set of primers for its ability to amplify the different species. And the outputs are the barcoding database and and then the second output of work package 8, or the aim, is to survey species on offshore structures using divers, settlement plates, and eDNA metabarcoding, and to identify non-native species colonizing these structures. The outputs of this is evidence of presence and absence of invasive non-native species on offshore structures, sites at which offshore structures are planned. And this will support models being developed by Sophie, who is speaking next. For those of you that don't know, a bit of background on environmental DNA. So eDNA is DNA that is released from an organism into the environment. This can be different environments. So you can look at marine water, you can look at glaciers, you can look at soil samples, and you can use it to do a lot of different analyses. So look at the diet and food webs, the biodiversity of an environment. And the sources of this eDNA are skin cells or mucus, hair, and feces. So two of the kind of ways in which eDNA is analyzed is quantitative PCR, which is species specific. So you develop an assay that targets a particular species and look for that species within the water sample. This is kind of finding the needle in the haystack tool. And the other method is meta-barcoding. So this is all of the DNA within this sample, and it's pooled, and it will detect the presence of all of the organisms within the sample. So kind of describing the haystack. So some of the benefits and disadvantages of using eDNA is that the benefits are it's a non-invasive technique. It allows for rapid detections, and it's cheaper and requires less manpower than traditional surveys. Whereas the disadvantages are that different environmental conditions can affect the breakdown of DNA. So for example, the amount of UV radiation or primary productivity occurring within an environment. And it also requires a reliable comparison database. Because you don't have the specimens, you need sequences that you can compare to identify them. So some of the results from work package 4, this was a study published, done as part of work package 4, in which they looked for the presence of Didymnum vexillum across around the Irish Sea, so in open and closed and artificial and natural environments. And they developed a qPCR assay which did detect Didymnum vexillum, while the metabarcoding did not. However, although it didn't detect, the metabarcoding didn't detect Didymnum vexillum, metabarcoding can be found multiple invasive species from this study. So it just shows that although it didn't detect the one they were looking for specifically, it can be used to detect other species, so kind of getting a broad range of what is there. So then moving on to work package 8. So for the environmental DNA samples that were collected in work package 4, 95% of the operational taxonomic units, so the species, could not be assigned to species level. And this is why we wanted to build on this DNA barcoding database as a reference database to kind of maybe identify some of these OTUs that weren't identified. And then also some eDNA samples were taken in work package 8 as well. So University College Dublin have done a lot of work collecting seawater samples from the sites that you can see marked here in stars. And then they've also done a lot of work kind of ground truthing this using diving studies, fixed camera video and ROV surveys. As you can see, there are different types of sites, so the planned wind farms, established wind farms, and then older structures. And all of these contain structures within intertidal zones. So some of the sample locations for ground truthing, they looked at the inshore structures, so Dublin Port, and nearshore structures, so the navigational buoys leading into Dublin Port, and the offshore structure was the Kishbank Lighthouse. And ground truthing these studies, they found 217 native species identified across all structure types, but only 3 invasives were found only in the inshore structure. And these 3 invasive species were the colonial tubeworm Ficupimbatus enigmaticus, colonial tunicate Didymnum vexillum, solitary tunicate Styla clava. So my work has been kind of doing the DNA barcoding. So if you don't know, it's a method of identifying species using a short section of DNA. And there are 2 main ways in which it's used. So you have your specimen, which you know it's identified, or you know its species. You extract the DNA from this, and then you get a sequence, which you upload onto an international database. And other researchers can use this to identify their. Or method 2, you have sequences which you don't know what species it belongs to. So this would be, for example, our eDNA samples. We just have water samples. We don't have actual specimens. And you compare these to this online database, and that gives you your species identification. So to collect the samples for the DNA barcoding database, I put out settlement plates across in different marinas around the coast of Wales and on the east coast of Ireland. And these settlement plates were very basic, just a PVC plate that was sanded down to allow the species to colonize it, with a brick that weighted it down and about 1 meter in the water column. We also put some of these settlement plates offshore on the Jim South Cardinal Buoy and on some moorings that we built in Gwyntamore Wind Farm and Moor. So we collected all the samples, and as you can see, there are these are the samples that we collected. You separate them out, and what you need for a good DNA database is you need to keep a morphological sample. So if anyone calls into question your identification, they can then go back and have the sequence which is linked to the sample and look and check it themselves. Then for our lab work to create the DNA barcoding database, we've extracted all the DNA, selected primers to amplify certain areas of the genome, so within the mitochondrial and the nuclear genome. And the genes we focused on were CO1, Site B, 16S, 18S, and 12S. And now we're producing the DNA sequences for all these specimens, and going to upload them onto an online repository for researchers to use as this reference for further eDNA studies. So just again, to go back over the aims and outputs. So the main outputs for work package 4 and 8 were the qPCR assay for Irish sea strains of Didymnum vexillum, the barcoding database, and the evidence of presence or absence of invasive and non-native species on offshore structures. That's all our collaborators. Thank you very much.